should be a login page.
So good morning again, everyone. My name is Rich Shilsky. I'm a medical oncologist. I'm the chief medical officer at the American Society of Clinical Oncology, and I had the real pleasure of participating in the planning committee for this workshop, which I think has gotten off to a terrific start. So I also have the pleasure of moderating this session, which is really the lessons learned session, or in some cases, the lessons being learned from ongoing studies or studies in development. But hopefully it will continue to stimulate much of the great discussion that we've already had. I think Mike Pakinowski's talk in the last session really laid a lot of the groundwork for much of what will be discussed here, which really relates to the more practical issues of how to use genetics and genomics in the actual design of clinical trials, the implementation of clinical trials, the recruitment of participants in clinical trials, and the analysis of study data. I don't have any introductory slides. I think you've seen and you're all familiar with the fact that certainly in oncology, we've had a number of successes already in this general domain, particularly with respect to previously very intractable cancers like melanoma and non-small cell lung cancer, but increasingly in a wide range of tumors where understanding the genomic profile of the cancer and developing a drug that is specifically targeted against activated molecular pathways certainly seems to confer substantial benefit on those patients for whom the drug is appropriately used. Now, of course, the challenge we have in cancer is the frequent development of drug resistance and the fact that in many cases these targeted therapies do not result in long-term disease control. Nevertheless, the paradigm has been, I think, well proven now in oncology as a way of initiating an effective intervention for many patients, oftentimes with considerably less toxicity than what we've been accustomed to over many decades with the use of cytotoxic chemotherapy. So in this session, we have two examples drawn from oncology and two examples from non-oncology areas, but I think in each case, some very exciting examples that will illustrate some of the key principles in how to use genetics in drug development. So we'll start off with Roy Herbst from Yale, who is the principal investigator for an NCI-sponsored protocol called the LungMaster protocol. Many of you may be familiar with LungMap, which is a genomically driven study to identify new drugs with activity in a particular subset of patients with advanced non-small cell lung cancer. So, Roy? Oh, and one other thing I might mention, unlike the previous session, what we'd like to do in this session is to try to just take a very limited number of sort of clarifying questions after each talk, but then leave time at the end for a panel discussion. So if you have a burning question that needs to be addressed to clarify a key feature of someone's talk, please feel free to get up and ask it. Otherwise, we'd like to hold the questions. We'll have plenty of time for a panel discussion with all the speakers at the end. Right.
Thanks, Richard, and it's really an honor to be invited today to speak at this meeting. So I'm going to talk about the first example, LungMap, which deals with lung cancer, the #1 cause of cancer death worldwide. So sort of a good way to start is how are we going to make things better? Well, when I was a fellow about 20 years ago, this is what lung cancer looked like. Metastatic disease presenting in more than 50% of the patients, and survival curves with platinum therapy. These were actually considered improvements. These were the new drugs, and of course, you recognize many of them. But you see there's very little of any tail of this curve. The survival at 1 year was less than half. So really, it was clear a new paradigm was needed. Well, I was very fortunate. I moved to MD Anderson at that time, and I worked with Ki Hong and John Mendelsohn, and they said, well, we have this new drug, ZD1839. Why don't we try this in the phase 1 unit? And we started treating patients with lung cancer who had bronchoalveolar disease like this. And in about 1 in 10, we saw that they got better, totally unheard of in metastatic disease. And of course, we've heard about this from the last speakers. This was an EGFR inhibitor. This drug went on to be known as gefitinib. Of course, there are now third-generation drugs in this setting. Patients do very well, but we didn't know back then why, and actually the drug became approved without a molecular correlate. Well, then, of course, we learned about the EGFR mutations and specific mutations that confer sensitivity to these drugs, and the entire world of how we treat lung cancer changed, and it actually changed enormously in the positive way for patients. So we started to think about lung cancer as a chronic disease, just like heart disease and diabetes. So this is Cecily. She was 9 years, one of the first patients on that phase 1 trial. Ultimately, she succumbed to her disease. Today, she probably would be able to get a new drug, osimertinib, which works in resistance. So the goal here is to keep patients alive, strong quality of life, as we find the new molecular targeted drugs to help them as they become resistant. Well, we knew we needed to do better with clinical trials, and actually in this very room, the National Cancer Policy Forum, maybe almost 9-10 years ago, created a committee, and we had meetings like this to sort of develop the IOM report. And of course, this was chaired by John Mendelsohn, who chaired the National Cancer Policy Forum at the time, really with a goal to improve clinical trials, bring more drugs to clinic, improve the speed, the science in the trial, the innovation. We knew this needed to be done. And coming from that, there were a number of concurrent efforts in lung cancer that tried to address this report. One was through the Thoracic Malignancy Steering Committee of the NCI. This was chaired by Fred Hirsch at the time. I was a member of that. But then there was this group from the Friends of Cancer Research Brookings that I chaired. And actually, we decided to take this report, and this was first presented at the Friends meeting, which all of you must know, led by Ellen Siegel and her team in 2012. And actually, we said, hey, this actually makes sense. Let's target lung cancer. Let's develop some sort of innovative master protocol, an umbrella protocol where large numbers of patients can be filtered in, find their genetic abnormalities, and then treat them based on that to develop new drugs faster in FDA-approvable ways. The challenge was that as lung cancer, of course, evolved with the discovery of the EGFR mutations, more and more mutations were found. But the question is, could you match those mutations with a drug? Most of the mutations that we hear about that are actionable are in adenocarcinoma, more common in the United States, about 60% or so. Many of these are never smokers. But there was very little at that time for squamous lung cancer. We knew a little bit about the molecular environment. So we said, what if we developed a trial for squamous lung cancer, unmet need, use some of these new trial designs? And that's the origin of the Lung Cancer Master Protocol. We really wanted to use broad-based next-generation sequencing. We wanted acceptable turnaround times, again, always with the patient as our focus, and how to get drugs more quickly approved, or learn that they're not approvable and move on to others. So you'll hear a little bit about this in this session. One of the next talks will be a basket, but this is an umbrella trial. Well, patients with squamous lung cancer, a similar type of disease is quite common. Lung cancer is about 200,000 patients a year in the United States. Squamous is about 30%. So you can imagine it's quite a few patients with very few options at the time that we did the study. The idea was to identify and test new targeted treatments and immunotherapies for squamous lung cancer, and if effective, move them to approval, and it was known as the Lung Map, and we ran it through the SWOG, one of the cooperative groups, and it's SWOG 1400. Well, here you can see why we chose squamous lung cancer. We were already screening, so that was good. 63% of lung squamous cancers, we could identify some target, some potentially actionable mutation. But we didn't know if a drug would work in that setting. It was an orphan disease at that time. Things changed a little bit as we were developing the trial, in that immunotherapies came into play, and you'll see we had to adapt to that. But now immunotherapy has become a major component of the MAP trial, and what we're actually going to need to do is figure out in those patients who become refractory to immunotherapies or primarily refractory, which are still a pretty large number, maybe we can use the lung MAP mechanism to now find markers not only in the tumor, but now in the microenvironment, so adding a complexity there. And certainly we knew that we needed to do more research. So we developed this public-private partnership, and it really is a wonderful thing because with the help of the Friends, helping us coordinate it together, working with David Woolley and the team at the FNIH, of course, Jeff Abrams, Shakun Malik, and the team at the National Cancer Institute, and Janet Woodcock and Rick Pasner and others at the FDA providing an advisory role, we pulled this partnership together and we pulled all the cooperative groups together, because that was one of the things that came out of the National Clinical Trials Network. Let's not have one group do a trial and another group do a trial. Try to all work together. Let's find the 10% of patients with an FGF receptor mutation and do a study and figure out if those drugs work. And that was the master protocol. Now, a big part of this partnership will be on my next slide, and that's going to be the industry partners, because that's where we're getting the drugs and actually a good deal of the funding for this trial. And you can see in squamous lung cancer, details not as important as just lessons learned, but you can see there are potential actionable mutations. Fibroblast growth factor, amplification or mutation, as I mentioned, but it's 5%. You know, I'd have to screen hundreds of patients at my site. I could never do it at Yale to find enough patients to ask the question, do these drugs work? PI3 kinase mutation, 9%. Again, couldn't do it on our own, but as a team, as a collaboration, working with industry, with trials that could be FDA approvable, that was the goal. So we created this sort of village, an oversight committee that's chaired by Evelyn Siegel and myself, an executive operations group, the co-PI on this, we work very closely together, Valley Papadimitricopoulos and MD Anderson, and you can see all the partners, and it does become, you know, a logistics issue, and I'll talk more about that as we go forward, but we're working together as a team. Here are the partners, and again, industry partners. We held meetings here in Washington, at AACR, at ASCO, to bring in the companies. We said to companies, you can come do the master protocol. We'll find patients for you for your drugs that have small percentage genetic markers, and we actually had plenty of interest. In fact, so much interest that we had to put together a drug selection committee, and you can see that it's a very diverse group of academic and industry and some consultants, you know, who were helping us out. And we put that group together so that they could advise us, is this target valid enough? And that's a big question. Do you have enough validation on a target going in to match it to a drug to put this into this master protocol? Now, of course, once it's in the master protocol, it's available throughout the United States, underserved areas, everything's paid for as far as the profiling, because all the profiling was being done by Foundation Medicine with some of the resources from the partnership. So here you have it, a multi-arm master protocol, homogeneous patient populations, and consistent eligibility from arm to arm. Each arm is independent of the others, so it's a modular type of design. Infrastructure facilitates opening new arms faster. One closes, another opens. At a phase 2-3 design initially, we thought we'd look to see if there was activity in phase 2, and then it could seamlessly transition to phase 3. We wanted to get drugs approved. We wanted to screen large numbers of patients from multiple targets, broad-based platform. Janet Woodcock, who was very closely advising us, constantly made the case this has to be a community trial. Most patients in oncology are treated in the community, not at the academic centers, probably more than 90%. So really one of the things we're very proud of is that we're getting this out to patients in the community, we're getting it out to underserved areas, provides a sufficient hit rate to engage patients and physicians, and bring the drugs to the patients. So here was our initial design as we first envisioned it and started it. Centralized biomarker profiling. I mentioned we used Foundation Medicine. We did put in an RFA, and we brought in several groups, and we picked the one that could do the best work at the least price. And then, of course, we had biomarker positive arms of screening. Within 2 weeks, you get a genomic next generation profile, and for those we had studies. And then we realized that even in our best of worlds, we'd have 30–40% of the patients who did not have a biomarker that was identified. And for those, we had a non-matched study. And at that time, 3–4 years ago, when we developed this, immunotherapy was coming into play. So that was immunotherapy. And as I said, each study is independent of the others. One could drop out. We can add a new one. The biomarker studies would progress to phase 3 if they met their endpoint. Well, we've had some evolution over the years. Again, I want to get to a few lessons, so I won't go through this in too much detail. But here are our biomarker-driven sub-studies, and the first 3 that we chose were a PI3 kinase inhibitor, a CDK4/6 inhibitor, and an FGF receptor inhibitor. And actually, what we've learned is all 3 studies closed, they fully accrued, and the drugs were not effective. Now, not the result I would want as an oncologist who sees patients. However, we've answered a question, the mechanism worked. None of these studies would have filled if we had tried to do them as single arms. Now the goal is to try to find the right drug or a combination of drugs so that we can see effect. We actually have 2 very exciting new arms about to open. Actually, this one just opened last week. This is a PARP inhibitor, and this is a c-MET antibody. This is an armed antibody, so it might not be that we have a genetic driver that we can block, but we can use that target to bring an armed antibody to play, and this is an AbbVie drug that's about to start. We're also very intrigued now by looking at checkpoint inhibitors, and these are combinations of CTLA-4 and PD-L1 or PD-1 inhibitors that we're studying. This is our new design. Again, time doesn't permit me to discuss it. I'd be happy to entertain discussions at lunch. We're now modifying the trial, and this is where we're going to be getting input from academia, from physicians in community practice, from basic scientists, and most importantly, patients, to figure out how we're now going to use combinations of different immunotherapies and target, different sort of populations. But in the last minutes, a few lessons learned. We learned in order to find patients, it wasn't enough to just screen at progression. Someone comes in, they have refractory disease, we heard from Jane. They don't want to say, oh, well, let's find your tissue, let's do a few weeks, we'll get you on the trial. So what we started to do is pre-screen. We knew all these patients getting chemotherapy for lung cancer were going to fail, so we actually added some resources and we started pre-screening patients so that at the time of their first chemotherapy, they could enroll in a pre-screening option for the trial so that we could get them on the trial when they progressed. We'd have that information available. So we added pre-screening and screening at prevention, and then either they had a biomarker, they went on a matched study, or no biomarker, a non-matched study. Look at what we've done in the several years we've been open. These are the sites that are open around the country. We just got Canada open. A little bit of regulatory hurdles there, but we got them open. 740 sites open, over 1,100 patients registered, and you can see the pre-screening is picking up. More than a third of them are being pre-screened, and 800 patients were assigned to one of the sub-study, and 436 are registered at this time. We paid well. In order to get this to work, we learned that we're competing with industry trials. These are, in many cases, industry trials, but the cooperative groups were not paying quite as well, so we actually had additional funds and reimbursements that we gave to sites. So again, the detail's not as important as we did try to provide nothing that was above what they needed, but enough to do the trial and support the research staff they needed. This is the key slide. When you're looking for genomically driven populations, even in a common cancer like lung cancer, imagine if you're doing this in an orphan-type cancer disease, you need a big funnel. You need a lot of patients, and we're screening through to get those 5%. Now we're even talking, you're going to hear in the next talk about a drug that's even less than that. So we need to find the patients. Well, one thing we did is we created an accrual enhancement committee. A lot of help from the NCI on this, from the Friends and other groups. We wanted to help sites that were already accruing. There are two ways to do this. You could get sites that aren't involved and get them on, or say, well, here's 700 sites, 400 of them are really good, or 300 are really good. Let's help the accruing sites. We also wanted to recruit new participants, and I now do Twitter, and we learned social media, promotional videos. If there's time, I'll show you one of those, outreach to lung advocacy groups and patients, and then extra support to accruing sites. So this is something that we figured out. If a site's accruing, help them. Don't waste a lot of time on the ones that haven't put a single patient on in 6 months to a year. Go to the sites that are highly accruing. We have a very nice website. You can see that here. It's really focused on patients. We're getting the word out. We have these types of flyers that we've given to the patient advocacy groups that are available around the country. The trial's available at multiple locations, trying to explain in more simple terms what it is, that you're matching a drug to the disease, a little about personalized medicine, a little bit about cutting-edge approaches. So this is a quick video. It's 2 minutes long, and with Rich's permission, I might go 1 minute over, but I'm going to show you the video that was prepared using a patient, a veteran. So so here it goes.
I was happy to learn that there was an option after the initial radiation and chemo.
A clinical trial offers new hope. It offers something better than the status quo and the potential to get a new drug earlier in a very well-supervised setting.
LungMap is a precision medicine trials that is trying to help patients with squamous cell lung cancer. And it is a trial where multiple drugs are available to the patients under one umbrella.
This is a public-private partnership. We have a large trial. We're funded by the National Cancer Institute, but also by a lot of funds from industry who have come together to work with us.
My doctor recommended I participate in the clinical trial, and I was happy to try it.
Precision medicine is when we have a drug that is found to target mutation in a patient's tumor. So it only will attack the patient's genetic mutation that drives the cancer, but not normal cells.
We need to find the drug that's best for your particular cancer. You know, what is it that's driving that cancer to grow, and then pick the best drug from the portfolio of drugs that are in LungMap.
Even if your tumor does not have a target for which we have a drug in LungMap, you may still be eligible for immunotherapy. Immunotherapy is a medicine that helps your own immune system fight cancer.
So in LungMap, you can get some of the most modern and innovative therapies. We're learning how they work, and we're going to continue to find even better drugs potentially in the future.
My clinic visits are very easy.
Patients can come in to one of over 600 clinics in the US and Canada.
Oh, getting to the hospital has been no problem at all.
Profiling in LungMap at Foundation Medicine and the drug therapy is all provided to the patient at no cost. The only way we're going to really make a difference is to all work together to take our best tools, find the best science, to bring those tools to the patient, and that's what LungMap is doing.
I feel great. I'm totally happy that I was chosen for this study.
This is personalized medicine at its very best. I should have just showed you the video. It'd be better than the whole lecture. But really, we had a lot of help, and a lot of help from the partners, and actually through the friends we testified before 21st Century Cures, and the policy issues are shown here. Biomarkers, diagnostics, partnerships, and resources. And really, this is one mechanism of making work. But even with all the resources, all the cooperation, it's been fraught with issues regarding accrual, finding the right drugs, finding the right markers. So that's why symposia like this are so important. Thank you very much.